All right, so hopefully I can get slides to move correctly. All right, excellent. I can work the pointer. This is the very first thing one needs to know how to do when presenting. So thank you very much for attending my talk. Those of us in this digital transformation space or the interface of IT and science, I think we have extremely challenging jobs every day. So I really like to start myself reminding myself with the why, right? So we really do this for the patients, right? Making sure that we're bringing the medicines to really make their lives better. And this is really AbbVie's mission, and the countries that we serve patients in, and the therapeutic areas that we act in. And I think it's always a good reminder for me of the why, because the days are really, really hard. So now I'd like to talk a little bit. This is a role that I had previously, but the Zippo team was nice enough to let me still present on an old, a little bit of an older story. But this is for the Bioprocess CMC development organization that I was a part of, and I led their digital transformation strategy and enablement for about 4 years. Before I took that role, I was supporting them as one of their key IT partners. So this story has a little bit of a longer timeline because I had been involved with this group for a little bit longer in different roles. The CMC development organization, I know different companies organize it differently, but at least at AbbVie for biologics drug substance CMC development, this is really end to end. So the team gets started as the sequence is coming from discovery, starting cell line development, and the organization also does all of the commercial on-market support for all of our products. So it is a wide range of understanding the molecule and the process to manufacture it and the analytics to control it. And the team, the majority of the team is located at our site in Worcester, where our biologics pipeline and launch site is, but we also have team members at all of our biologics manufacturing sites supporting those ongoing things. So we have Singapore, Puerto Rico, and our sites in Irvine, California, and our newest team in Waltham, Massachusetts. And again, the biologics CMC development is all biologics modalities, so these are increasing in complexity and scope. So you can see the scientific challenge of this organization is quite a large one to solve. So I'd like to start a little bit with the approach that we took to digital transformation. And when I joined the team, I wanted to make sure we started with our why. And in the CMC development space, there's a lot of talk and discussion around prior knowledge, and everybody's focused on prior knowledge and how do you use prior knowledge to move faster and things like that. And so how we anchored ourselves was saying, hey guys, the data that we generate in the labs, or the data that comes from our QC labs or our manufacturing sites, that's an asset, right? And we need to treat that data like an asset. So we have to make sure we have the right context around it. We need to make sure that we're doing structured data capture, not just throwing this in a file that nobody can read. Well, now AI can read it, but that was not until recently. And then making sure that we're sharing this data, storing this data in a way that it can be not only used, right? So choosing my control strategy, setting my method parameters, but also reuse. So use that exact same data for my BLA filing. So really making sure that we're setting up our data systems for that primary use and all of the secondary reuse we have for it. And so as an organization, we had plenty of data, like data coming out of our ears, right? Many of it, these are actual pictures of labs, stacks of papers, like thermal printouts off of instrumentation, printouts out of PI, paper upon paper upon paper. The vast majority of the lab data is in Excel files and PowerPoints on network shares, or in many cases, my scientist OneDrive or our drive, right? And of course, we have plenty of data, so in our manufacturing spaces, our data is structured, but none of that's connected to anything else, right? So any insight or things I want to do, I have to go and transcribe data. And where we wanted to move to, and I think that's where we are today, is really what we call the fit for science integrated digital ecosystem. So find the right systems and tools that work for that scientific instrument or that way of working or that type of experimentation, and then make sure we're connecting them and integrating those systems so that the data flows. And so the way that we went about this as an organization was to really start with champions, right? Who are my scientists in the lab, the SMEs, right? And it's not just about digitizing the data, but it's also about simplifying the workflows and changing how we work. So it's not a digitization process, it's a digital transformation process because we want the scientists to change how they work. And we really needed to have co-ownership and real partnership between our scientific teams and our IT teams who are delivering the technology. And so for the software developers in the audience, this graphic is insanely overused. However, for my scientists in the bench, this was actually quite good, right? We kind of said, hey guys, this is not going to be perfect, right? We are going to do a co-evolution of our technology tools and the way we work in the lab, and we're not going to get it right the first time. So it was really great when I started to have scientists saying, hey guys, remember, we're on the skateboard, but we're learning and we're getting better, right? And so we really had this commitment as an organization to say it's not about delivering this one piece of technology, but it's about delivering this use case and the end-to-end data that makes up that use case and the workflow on the use and the reuse and things of that data. And so now I want to just dive a little bit more into the actual systems and data architecture that we had. So we generally, this is a one-way, but it's a little bit more complex than a one-way, but we generally had the instrument data, and we bring that then into our experimental context and workflow management layer, because an Empower result is kind of useless unless I know what was all the experimental conditions that generated that Empower result. And then we bring this into whatever your data platform, your data warehouse, whatever the term of art is of this day, and then enable data visualization and data insights. And more traditional projects that I had experienced in the past would be deliver the limbs. And what we were trying to do is deliver the end-to-end. So yes, I deliver the limbs, but I also deliver the integration with the instrument and the data visualization that you need for that. And really, this was really trying to make sure that we had all of this data at our fingertips, so getting that near real-time data available for the organization and minimize the amount of manual transcription and things that we're doing. But again, the end goal was retain and repurpose knowledge by making sure we had this foundation strong. So now I'd like to take you through a little bit of a journey, an example of our analytical lab. This is quite a long journey that the team has been on, but I'm really proud of the team and how far they come along and the efficiencies that they've gained along the way. So we started, I'll throw an imaginary date in there. We started building ELN templates in 2017 or so, and the team was also getting more and more analytical samples coming in, so we built a SharePoint site to help them manage the request fulfillment process within the labs. I'm sure many of you are familiar with this. About the same time, we started bringing in lots of high throughput instrumentation to handle more samples, to handle more results coming through the lab. For us, we realized the ELN templates weren't going to be the right solution for us, so we pivoted to a LIM solution. We had some people who thought, oh, this is a CMC development lab. Your data is going to be used in a filing. You must have a GMP IT system. Which took us a little while to then go, no, we don't actually need a GMP IT system for a non-GMP development lab. So we pivoted, and as we pivoted to our non-GMP solution, we delivered that end-to-end vision. So as we delivered the limbs, we had the integration with Empower and the Spotfire visualizations as the package that we delivered to the organization. And since then, we've gotten more instruments, more two-way integration with our high-throughput instrumentation lab. We've made improvements to our SharePoint logistics system that everybody uses to track the samples, because there are tons of samples going into the lab and data coming out. And so what this kind of looked like is, if I think about the ELN days, yes, the analytical results were digitized, but it was all done manually. All of the analytical scientists were manually transcribing from every different instrument and then putting it into their ELN. And the way that we had our ELNs set up is I had the analytical scientists putting the data in one ELN and the requester, the bioprocess development, having a different ELN. And the ELNs talked, but there were sometimes problems. So everybody kind of double-checked and triple-checked all the data. So it was digitized, but it was highly manual. And then to get any graphics or things that they wanted, they would then take it out to their Excel file to make their different graphics. So highly manual process across the entire organization. As we kind of re-anchored, we integrated our bioprocess development solution we called Espresso. I'll tell you a little bit more about that. We have a direct integration to hand off the sample data and the analysis that are being requested into LIMS. We have the integration, two-way integration with Empower. That's about 80% of the team's data. And then with the high throughput automation for sample prep. And we're continuing to get more and more easy data off of the instrumentation. So at a minimum, it's get an export with your sample IDs, take that to your instrument. Now you have your file format to upload back to LIMS. So it's not fully automated, but it's as efficient as we can get it in a manual process. And then all of this data is available within Spotfire dashboards of just the analytical data, or we're also pulling back the analytical data in the context of the bioprocess development software. So you've got your manufacturing conditions and your analytical data in a single spot. And along this way, right, it's not just a lab digitization effort, but we also brought in all of this high throughput automation. So we're getting better results because of that automation. We're getting it more efficient for our lab scientists to use it. At the early part of the ELN days, we were still on standalone chromatography systems. So moving to Empower just about as COVID hit was actually a good thing, right? You could see your data from wherever you are and analyze it. So a lot of benefits to the overall organization. However, quite a long journey to get there. So now I'd like to talk a little bit about our process development labs, so cell culture and purification development labs. And I know there was conversation this morning about build versus buy and pros and cons. In this case, we went with a build solution, so I'll tell you a bit about that. So within any experimental data system, I often think of this as a data model. And it's like multiple competing data models for scientists. I need this data model to mimic the workflow that I do in the lab and the way that I work in the lab. But I also need this data model to mimic the actual science that I'm doing in the lab. So it is a quite complex data model that you're trying to hold both things together. So if I just give you an example here, we kind of have a project, an experiment. We have the containers that we're doing the experiment in. We have the batch, which is kind of the material, so the cell culture or the purification that we're doing. And we have inputs and outputs, so measurements or media or solution that we're doing into this. So how this looks for a given cell culture experiment is I may have my vessel or my well, and this could be a variety of things. This could be a 96 well plate, a single well. This could be a flask. This could be a bioreactor. It could be a wave bag. It could be a 200-liter bioreactor in a pilot lab. And we have the cell line that went into it. We have the medians, the solutions and feeds that went into it. We can take samples out of it, and we can put measurements on top of those samples. And so the other thing that happens in a bioprocess development lab is you're inventing the manufacturing process. So in manufacturing, I know what I'm doing. It's the same thing every time. But in a process development lab, I don't know. And so the way that our LIMS work, one of my colleagues called it a choose your own adventure LIMS, right? So I don't know, I don't have a set workflow I'm always doing. And so what we have is a directed acyclic graph concept that allows us to let the scientist take the experiment where it needs to go for that given experiment. And each of these kind of colored graphics is an entity. And each entity can have different business logic and context and metadata that make sense for that entity. And we have specific ways that those entities can interact based on the scientific and workflow data model. And so this is an example where I've taken the pink is 4 different cell lines, and I'm doing them, growing them, and then taking some measurements. So this is like a seed train experiment. Here's maybe a more traditional seed train N-1 bioreactor and then a bioreactor production experiment. So you can see the lots of samples coming off from the production bioreactor sample. So the scientists can see graphically as they log into the system, what did this experiment look like? And you can kind of get that context almost by just looking at this graphic. And so just digging in a little bit more, this is our vessels. We would see that wave bag, that bioreactor. We could see if it's a seed train versus a production bioreactor. I have to see what this one is. Oh, aliquots I can pull, and I can know exactly which day I pulled that aliquot from. On the measurement runs, I can say, hey, for this sample or this aliquot, these are the measurements I did. So this could be my bioreactor conditions, it could be my Visol results, it could be my osmometer or my ABL. And then each of the measurements would have the results. And so it was actually really hard for scientists who had been in an Excel file and putting in this way to start understanding why you needed to put the data in a different way. But what this allows is now we have all of this data in a data model that we know how it interacts scientifically, and now we can very easily get the graphics and get the visualizations that cell culture scientists are used to seeing. And so I would have my typical things that I'm looking at in a production bioreactor. I can say, oh, I want to see this experiment and this experiment overlaid, or I only want to see that one, or knock those things out. So getting here was hard. Once we got here, I think they really started to see the value. So now let me take you over to the purification development. So it's the same directed acyclic graph, same concepts, but now our entities are different. Our entities are really unit operations that occur, right? And again, a purification development experiment can go off in many different directions. So this is, one example where I've got a couple chromatography steps and then I'm taking some measurements. Here's a more complex one that I wanted to see how different, if I took it this path versus that path, like what would give me the best downstream process for a given molecule. So I have to come over so I can see. So within chromatography unit operations or within filtration unit operations, we would have those steps that occur within the unit operation. So within a chromatography, I would say, this is my load, this is my wash, I'm tracking which buffers I'm using, I'm tracking my column conditions. In an ideal world, I'd love to pull it from ACTA, but we didn't quite get there, but you guys get the point. We have automated yield calculations, so they want to know the yield from a single unit operation, but also the yield of the overall unit operation train within the downstream process. And then a lot of the analytics for the purification team are actually sent to the analytical team. So then we're tracking, hey, I'm sending these samples over and I want all of these measurements. And so here's an example of a visualization where they wanted to compare their impurity clearance in their development runs and then also their rep batch runs. And so pulling this graphic together, you had to go across 3 different teams, across a couple different experiments, all agree to put your data in the same space. Now it's all there whenever anybody wants. And so kind of a ROI, I guess, if you will, is for the cell culture team, like this was very highly manual, lots of thermal printouts from lots of different instrumentations. People would walk around with clipboards in the lab, writing everything down, and then they'd transcribe all of those clipboards to an Excel file. But that Excel file was set up for the daily sampling, not for the way I wanted to view the data, so you transcribe it to another Excel. You guys get the point, highly manual. Now, right, we're saying, okay, no, design your experiment in Espresso first, set up your bioreactors. We know the sample ID is coming off, put that information onto your instruments. We have a semi-automated pull into Espresso. We have an automated pull from Pi, so I can say, click a button, give me all the bioreactor conditions at this time, this day, and they're pulled in automatically. And then again, out to Spotfire and Pi. So we put this together, this time savings, a couple of years ago. But really, if we compare just the documentation, so all of the transcriptions, all of the double checking of the work, for a 2-week bioreactor experiment, so just said that, 50% of a scientist's time, roughly, was spent on documenting this experiment. That's now down to 15%. And I didn't have a chance to update the slide because it came out after I left the job, but They said now started doing this for PC studies. So for a process characterization study, I'm pulling many, many different experiments. And how they used to do it was get everything into a single Excel file. Somebody else come back and confirm 100% of those data points are correct, and then I can go off and do my statistical analyses and calculations. So that pulling together all the data into a single Excel file, reviewing 100% of the data points would take at least a month, right? That's now down 2 days because of all of this data automation and the way that the system is set up. So the organization is, as painful as it was to get in the system, is starting to see those efficiency benefits. And now lots of data to build machine learning models, to do different ways to do their process development work. So I'd like to end a little bit with what I think we as a team figured out as our success factors along the way. So the first one for me is really co-ownership of both the strategy and the execution between the scientific team and the IT team, making sure you have dedicated individuals in both teams and aligned vision and clear prioritization. I think the other big thing for me is organizational readiness for change. So my VP said, hey, this is a great place to go. I said, we don't have the right SME, and that team is slammed right now. So no, we're not gonna go there. Once I have the right SMEs and the team can say that they'll take the change, then we'll go. So we waited a year, and then it was successful. If we'd tried before, it wouldn't have been a successful thing. So, and even when the new way of working, everybody agrees it's gonna be so much better, you still have to learn how to do it. And you're gonna take a little efficiency hit along the way, so you have to acknowledge that as an organization. There are many big and small decisions along the way, and there's no right answer. There's multiple right answers and there are multiple wrong answers. So in the small things, you've got to enable the team to make the decisions quickly, right? And you have to be willing to pivot when required. Do that carefully. And really, to me, I loved the morning talk saying the 80-20 rule. Absolutely, 80% solution. You're aiming for the 80% solution. And ideally, as an organization, you're committing to continuous improvement after that, right? And so, to me, it really having a continuous improvement mindset, but with the big picture and the long-term vision in mind. If you have grassroots continuous improvement, it'll go off in 100 different directions. So try to harness that and go, this is the long-term where we're trying to go. Let's harness that energy so that we can make lots of small progress along that way to that big picture vision that we're going. So I do want to, this was a multi-year effort, a very large team, much larger than those I'm acknowledging on the slide, but I did want to really acknowledge many of the scientific leads within the various teams, as well as our really phenomenal IT partners that we have at AbbVie. With that, I'll take any questions. Yeah.
First, I want to congratulate you on the exemplification of the process. That's a major step on its own.
I still have the scars.
I was interested when you were talking about the different data models in terms of business processing and scientific, and then you referred around data consumption or analytics. Have you seen, by visualizing this for the scientists and you, have you seen actually changes in those processes in terms of why are we doing this in the business process and the scientific process?
Not really yet. Where I would say we've seen it is I wanted the adoption. And so we didn't add a lot of controls around how they entered the data. Because what I knew was gonna happen is we would go round and round around about how you call this thing, and then we'd never start using the system. And so we kind of let it go in dirty at first, and then as soon as they had the visualizations, they're like, but I can't compare experiment X and experiment Y. And it's like, because you didn't call it the same thing. And then we went back and started adding in more data standardization. So that would be, I'd say, the change that we saw. And that was a strategic choice we made that was to push for adoption rather than getting data clean at the time.
How did you manage historians and time series data?
Not well. No, I think we did have an effort that I didn't really share, that we did get all of our all of our 100-something bioreactors connected to Pi, so it is there. I think the folks that have come in after me have a lot more experience in that space, so I think are focusing on that now. But it is challenging to bring together that really continuous data and the more static data. I've not really seen it done well, to be honest.
Excuse me, you can ask you a question that maybe we can actually record it because
I think I have two kind of questions. The first is, how'd you get the XR data out? Is it still manual, or how are you getting that flat? I still struggle with XR data pretty heavily in like 3 sites now at this point.
You see that I kind of glossed over something. No, I'd say, and my new role, we have a slightly different strategy. In that space, we were just like, we're just gonna brute force Python script the files.
Okay, that's what we're doing now, so that makes me feel better. That's what.
I think there's a gap that I see in like the instrument middleware layer that I think we're all struggling with.
And the second, like this Espresso seems like a pretty cool tool. A is selling it, and B has your manufacturing side. Kind of everything you did on the non-GMP side is going to work like for CPV reports and whatnot on the GMP side. Have you guys looked at making this more on the manufacturing floor? But still Pi and XR, it's the same stuff.
So, I mean, we very strategically said this is all non-GMP. These are all non-GMP systems. GMP, go pick whatever system works best for you. And now we're at this point where they have their data systems, we have our data systems. So now we're trying to say, okay, how can we bring our data together? Where are the gaps? And like, let's not try to share systems, but let's try to share the data.
Makes sense.
Hi. So I love the fact that you pointed out organizational readiness. That's like, we never have enough conversations about that, but for you to kind of elaborate further on how you got that, you got it delayed. Like, how did you navigate that?
So when I started the role, I part of what I did, and it sounds silly, but like I brought in some smart sheets for like workflow automation to show people there are like low code tools that could make your job easier. So then I got a little of trust with the organization for what I was saying. So when I was ready to say, guys, you're not ready here, there was enough trust build up in what I was doing that they listened. Which is a really non-answer, I apologize.
I think it always depends on the executive, like if they really want it today versus tomorrow, and what their trade-offs are.
And I'm actually doing that a bit now. So my role today is for AbbVie's QC labs globally. We have, I think, 22 QC labs globally, and they're all in different levels of digital maturity. So we're starting to have those conversations with the site leadership and the senior VPs and go, yeah, I know you guys want your labs paper free, but we kind of need you to do X, Y, and Z first, right?
I guess I'll go next. I mean, I'm always interested in a little bit of the how after the why and you defining the what's. So you talked about having like just something with, sorry, I forgot what I was going to say about that. Small decisions versus large decisions. So I'm coming from a global perspective where even a small decision, like on, for example, like trying to harmonize a term becomes like a governance project and meetings and everything. So I just wanted a little bit of insights from your end on how would you look at a small decision like that, and did you start with like one site and then kind of build that up and then go global as you just mentioned, or did you have to revisit stuff as you did that as well?
So the way that the Bioprocess organization was structured at AbbVie is the vast majority of the team was all located in Worcester. And so that was going to be the consensus decision that would then be adopted by the other sites. So we had, I guess we didn't have some of those constraints that we had to fight through. As we've moved to the next sites, we've kind of said, yes, we'll allow you some freedom here because you're a smaller team and you don't need this, but this is very important for the data model and the data use. We're not going to bend on those things. So it was a small enough team, all co-localized, that we didn't have quite those challenges.
From my experience, that is the way it's for success, is to like have a localized site as a lead site, yeah.
You can get burned by that as well.
Connecting data has been a problem now. Yeah. All right. Thank
So,
you.
Yep.
Pretty impressive stuff with espresso. You said you went through determining build versus buy. Like, how did you make that decision? And you said mostly localized to the, I think it was the ABC site
Yep.
in Worcester. Like, obviously, as you continue to scale, like, how are you thinking about that? And what does V2 of that whole process kind of look like?
V2 is no longer my call. It's somebody else's. No, I would say we did originally, like we had started in ELN templates. We kind of said, oh, ELN templates we don't think are going to work for us. We did look at some of the R&D type LIMS vendors. We did get, quite down the road on traditional requirements, documentations, and things. And then our leadership saw the dollar amount and the timeline to actually deliver that thing, kind of did an assessment of, oh, we could just build this, and so that was a bit of it. I think the landscape has maybe matured a little bit from when we started, so there may be other things that are available commercially that weren't there 5 or 6 years ago when we were making these decisions. So, and I only wanted one build. I did not want multiple builds in my ecosystem. So. Thank you guys so much.
Thanks, Dana, for sharing your learnings. We'll take a 30 minutes coffee break.